but obviously we have been together for 2 years now, so these students that we're here for are part of the biotechnology program. They joined all as sophomores and were with me as their junior and senior year. Funny enough, I actually recruited them before masks were a thing in February of 2020. So it's kind of an interesting story. I think these guys will always be a part of me because we kind of made it through this whole time together. And it's funny that now at the end, the last month and a half here, we're able to see their faces outside of the masks. So I really enjoyed that. But, we got to know each other with the face on our eyes, for sure. So also with these students, I'm incredibly proud of them. So I think we should say first, before we get into that, little ground rules. We have some water refreshments in the back if you ever need that. There's water fountains in the back as well. Restrooms are going to be out the back door. If you take a left, it's going to be on the left side of your hallway. It says boys and girls, but they're big enough for us for everyone, so it's okay. We are in elementary school, but I do really like this space, and the students, really get a good chance to show off what they've been working on this past year. So going back to the students, I am really proud of them. This is probably one of the most ambitious group of students I've had. Especially they come in every day talking about how great colleges are and how great this college is versus that one. I want to go to grad school there. I want to go to med school here. And it's not going to be okay if I don't. Realistically, that's fine. It's all talk. But at the same time, to have students that are that have those goals and that have that achievement thus far, I'm really proud of them. So without further ado, I'll hand over the mic here. But I really hope that you guys enjoy this. So they've been working on this Capstone project all year. Created an 8-page paper, at least 8 pages. I think the longest one is 19, but that includes an abstract and a title page and things like that. But regardless, obviously no small feat. But we're not going to listen to 8 pages of paper. What we have done is basically a very traditional science talk. And if you're not familiar with science, we have researchers that come in and they give a 5-minute talk about what they've been working on, what they use, what their results are, and what's next. So that's what we're going to see today. Most of them are going to be about 5 minutes because we have an hour and there's only 6 groups. They may take a little bit more, but regardless, they have practiced really hard. We've been working on presentations since day 1 of the program, so since last year. But regardless, I told them that they couldn't read off slides, and they kind of joked that, it's okay, but it's not. You will see, and keep them honest, you will see they're not going to read off the slides. All right? So we have Nathan and Aidan coming up.
making the fuel and stuff around that. But Ron was going to help us with our research. When we first met with him, it was through a Google Zoom, and he helped us sort of like go on track of what we wanted to research, sort of explain to us what we wanted to do, what we needed to research, and he also gave us some articles which also helped us out a lot.
We chose Biofuel because when Sergi was explaining what to do for the Capstone project, it jumped out at us the most. And we were like, oh, that's super cool. And we should really look into this since that could be potentially the fuel source of the future. We knew a little bit before, mainly from biodiesel, since that is actually made instead of biofuel made from algae or something like that. We looked up and researched a few different categories that you could potentially make biofuel from, which is cyanobacteria, algae, soybeans, corn. There's probably a few more that we haven't really gone in depth into researching yet. So far, the most promising source of biofuel is probably algae.
Alright, methods that help us along with our research. One of the methods we use is PubMed, which is like a database with a bunch of articles that have been peer-reviewed. So we use a lot of articles we got from there to help us along with our research. And as I mentioned before, Dr. Les provided us with some scholarly articles, which we also use in our research.
So mainly in algae, since it is a cellular organism, it's easier to use in science and manipulate, so we can use it for our own benefit. It's also the most researched potential biofuel that we have, and the second most research is probably cyanobacteria. We are looking at this so we can synthesize the lipids inside of the algae, which is what we would be using as the actual fuel source in and of itself. Since we can't really use any of the energy produced from any of the other organelles and all that inside of the cells of algae.
Alright, and then the other one we also researched was cyanobacteria. It's multicellular and it's a little harder to manipulate, that's why there's not much research going into it. But the research that is going into it, they want to use it because it harnesses the power of the sun, and then it's also concentrated CO2. And it acts as a biocatalyst, which is like a little mini factory, so it's also producing its own energy, which then can harness, which then we can bring into the biofuel.
The final part that we were looking at that's most heavily researched is biodiesel, since we can create it from using cooking oils and all that, like olive oil and canola oil as the two biggest probably producers of biodiesel. And in the process to make biodiesel, we get a few products, but the two biggest ones are the biodiesel in and of itself, as well as glycerin, which we cannot use. The biodiesel, we have to separate it from the glycerin using a specialized tool that is a lot easier to use than a micro pipette, as we'll go on to explain later on.
Alright, and then because we did biofuel, and as we mentioned, we can make biodiesel, so along with our Capstone, we actually made some of our own biodiesel, and this is sort of explaining what we did. So like first, the triglycerinate is like the oil we used. So we used that oil, and then we took the oil, and sort of warmed it up, and then when that was warming up, we took methane and sodium hydroxide, and we mixed that together. And it gave us methoxide. And once that was done, we took the oil and mixed it with the next oxide, which then gave us our biodiesel right there. And after all of that, we had a byproduct, which was glycerin.
So here we can see what our biodiesel was when we placed that inside of the flask. On the right side, we have two different types of biodiesel, one made from canola oil and one made from olive oil. On the other side, we have the glycerin that was the byproduct from there. In the left flask of the glycerin on the bottom, we have the sodium hydroxide, which did not fully dissolve into the solution of fatty esters.
Alright, and as a result, we actually got to test our own biodiesel, and it did work. On the right is it burning in a makeshift, I guess, cup, we call it. And then there's a little video we made of it sort of burning inside a ship, a little ship. You can see it burning, like there's little pop-up boats with like little ships, and then there's a candle wax, there's candle wax and that little silvery gold ladle that we lit, and then above that is a sort of little lake of water, and we will propel the boat, but sadly we couldn't get it to propel the boat, but we did get it to continue to burn.
What we learned in this experiment is biodiesel sort of has to be diluted in order to ignite, so we have to use wicks inside of the wax, which would soak up all of the biodiesel to spread it out a little bit, glue it, and then it would ignite.
Alright, and then there are some limitations to biofuel. It's not perfect. One is trying to get cost. Right now it's sort of very expensive to produce it. And then there's also mass production. Trying to mass produce it is kind of hard. Trying to figure out how to grow so much algae or cyanobacteria, all the different other biomasses. So we're trying to figure out how to really do that. We're trying to transport it and make it out so we can actually use it, and sustainability, refine it, so we can actually put it into cars and have it run.
What's next for me is I love science and everything it is, but it's just not for me. So I plan on going into an apprenticeship for plumbing and potentially starting my own construction business. I'm not going to go into the entire division because there's a lot in there.
Alright, and as for me, I'm going to Kent State where I'll major in biochemistry, and then I'll go on probably the pre-med track. And thank you for listening.
Next up, we have Anna Green, working on a chip.
So hi guys, my name is Anna Green, and for my senior Capstone project, I research human working on a chip. So a little bit about me, like I said, my name is Anna Green. I am a level 2 student at the Biotechnology Academy, and I'm also a senior at Stonebrook Falls High School. So I originally set out to learn what exactly working on a chip was, including their mechanical makeup and how they're able to function and keep living cell tissue alive inside this little in vitro chip. And so my original question went off of that, which was what are the beneficial factors of this chip and how possible is this in the face of animal testing in the future? And so although I did not already know much about human RNA chip, I did know that scientists had been looking for alternatives to animal testing, and it's a really highly talked about topic, so there's been a lot of research going into that. So for my research, I used 3 main methods. My first method was using PubMed and NCBI. Those are medical databases that have articles on medical sciences and biotechnology. I use that mainly to research the mechanical makeup of the chip and also like the RNA information of the chip. My next resource was GWAS Institute. So they did a lot of startup research on human organ on a chip. So they gave me a really great background information on the chip and just kind of what was argued about it also. And then finally, my major discussion. So it gave me a really professional view on how possible organ on a chip is in the scientific community and why it would be important to that. So an introduction to my mentor, her name is Dr. Aidan, and she's a molecular biologist by training. She's currently working for the EPA and focusing on toxicology, chemical exposures, and environmental justice. So although she has not worked directly with human organ on a chip, she has heard about it before and has worked on animal models that were a basis for it, mainly working on cell-to-cell and tissue-to-tissue interactions. So my first section on my capstone was the mechanical makeup of organ on a chip, and pictured is the mechanical makeup of the lung on a chip system. So there are four main components, including microfluidics, living cell tissue, drug delivery, also known as stimulation, and sensing. The microfluidics are what allow for accurate deposit of the pharmaceutical products inside the chip. In organs on a chip's case, it uses fluid shear force microfluidics, and that's what really allows for accurate deposit and release discharge inside the chip. The next component is the living cell tissue. Most chips are composed of a 2D cell tissue alignment, so that's how the organ cells are aligned inside the chip. The next thing is drug delivery, also known as stimulation, and that's what allows the cell tissue to have an accurate environment of what's inside the body. Some examples of that are physical or chemical exposure, electrical stimulation, and pulsatile flow. And then the final thing is sensing. So that's what allows us to see how the environment is going inside the chip, and it just kind of allows us to look into it in this little isolated microchip. My next section was the pros and cons of the is that it replaces animal testing in the science field and using any living organisms for experiments. And then it also allows us to have a direct window into the cellular process, and it happens all on the spice of the chip in an in vitro environment. Some cons of the chip are effectiveness. So right now, we are currently not able to use organ on a chip for anything involving the nervous system. So this kind of allows us to, it stops us from testing anything that involves pain management medications. And then also patents on the device. So currently there are multiple patents on different components in the device. So this allows us to, or this in turn regulates the developmental research of the chip and obtainability of some components in the chip. So right now there are currently 15 different developed human organ on a chip models. I focused on the three mainly highly developed ones, which were liver on a chip, lung on a chip, and heart on a chip. Liver on a chip is used to test toxicity of drugs and its impact on the liver. Lung on a chip is used to test environmental nanoparticles that we're exposed to on a daily basis and how they affect the tissue lining in the lung. And then heart on a chip is used to test for lasting effects of cardiovascular disease in the body and how it impacts the heart. And then my final section of my capstone was the future of the technology. So the first thing is human body on a chip. So one of the problems of human organ on a chip is it only tests one specific organ at a time. So by having human body on a chip, you're able to test how these pharmaceutical products go through the body and not just one singular organ. The next is angiochip. So angiochip is a biodegradable microgenome scaffolding which is implanted inside the body. Once it's implanted, endonucleation occurs, which is the covering of endothelial cells. And then after that, blood perfusion will go through the chip, and that helps with the healing inside the body. And then finally, microrobots. So right now, the fluid shear force microfluidics do allow for accurate deposit of the drugs inside of the chip, but microrobots would give us an even higher rate of accuracy inside the chip, and also gives a better rate of utility of the pharmaceutical products and drug efficiency. So if I kept researching human organ on a chip, I would definitely deep dive into every single model of human organ on a chip because they all have different components in them that allow them to keep the tissue alive. And then some other applications are using it to test for diseases in doctors' offices. So right now, human organ on a chip can only be used in specialized labs that were made to produce it. So another application would be use it in an everyday setting like a doctor's office. And then finally, my next study would be to really learn about other alternatives to animal testing, not just human organ on a chip. Some examples of that are molecular docking or QSAR assays. and me, majoring in animal science, and hopes to become a veterinarian. Thank you.
Next up, we have Ava Gantz from Hollis.
This one? Hi everyone, my name is Aidan and I did my Capstone project on Genome by Association study, otherwise known as GWAS. So a little bit about me. My name is Aidan Mathis. I'm a senior at Theodore Roosevelt High School, and I am level 2 in the biotechnology program at Biotech High School. At Kent, I am in National Honor Society, and I am also a 4-year letter in soccer. So first, the aim of my paper, after doing genetics in class, I realized that I really wanted to do something with genetics because I enjoyed the topic, and I wanted to evolve that somehow. I chose to do GWAS because I wanted to make a connection between genetics and medicine, and I also wanted to incorporate breast cancer somehow because my mom was a breast cancer survivor. And then my goals were to explain GWAS in depth and become an expert on the topic, and I wanted to spread awareness on the problems in genetics as well. So first is my methods. For my paper, I used 2 main methods, and the first one was government databases. So that includes PubMed, NCBI, NIH, and Genome.gov, and those are hubs for all scientific information where I got a lot of scholarly articles. My second method was meeting with my mentor. I met with my mentor back in January, and he and him discussed something called polygenic risk scores, which I will talk about later on in my topic, but he was very passionate about it. He also helped me narrow the scope of my project, as well as talk about the huge diversity problem in genetics. So a little bit about my mentor. His name is Dr. Aaron Wolf, and he got his PhD at the University of Washington, as well as doing a postdoctoral research fellowship at Princeton University. Now he works at the Federal Judiciary Center as an AAAS Science and Technology Policy Fellow, so giving them research on genetics and science. So first off, what is an SNP? An SNP stands for single nucleotide polymorphism, and an SNP is a variation in one single nucleotide. So nucleotide is the letters that make up your DNA strand of the A, T, C, and G. And an SNP has to happen within 1% of the population to be considered an SNP, as well as SNPs vary based on your region and your ethnicity, which I will bring up a lot later in my project about the genetics problem. What is GWAS? GWAS stands for Genome-wide Association Study, and a GWAS starts with 2 groups of people, one that has the disease that they're testing for, and another group that is similar but does not have the disease that they're testing for. DNA is then extracted from those 2 groups and sequenced looking for SNPs that are associated with the disease. Then they collect the data and compare side by side and eliminate the shared SNPs, leaving only the ones that are associated with the disease. And this is a GIF kind of showing how it happens. So there's a threshold line that eliminates all the common SNPs below the line, and above the line is the disease-causing SNPs that are left after. So next is the sections of my paper. In my paper, I divided it up into 4 different topics. The first one was the history of GWAS. The second one was breast cancer-specific GWAS. The third one was benefits and limitations of GWAS, and the fourth one was the future of GWAS. So for my first section, I did the history of GWAS, And GWAS was inspired by two different projects. One was called the Human Genome Project, which was a collaborative effort to sequence the entire human genome, which is about 3 billion base pairs. And then the second project was the HapMap project. And the HapMap project shows the locations of the SNPs in the genome along with the Human Genome Project, and it provides you genetic variants that could give you information on drug response, health, and developing disease. And these two combined could make a very good tool like this. My second section was breast cancer specific GWAS. So in breast cancer GWAS studies, they identified two main genes, which are called BRCA1 and 2. And BRCA1 and 2 stands for breast cancer gene. And BRCA1 and 2 are tumor suppressor genes. And when a mutation is on one of those genes, that is the SNP that they identify with the disease. And that's why genetic testing is so important in today's society, because you will look for those SNPs so that you know if you have the association or not. My third section was benefits and limitations of GWAS. So I covered many benefits and limits, but I wanted to talk about only one here for each topic. The benefit that I chose to talk about was that GWAS has many clinical applications. So this is talking about how it can be beneficial in prevention and early detection methods, as well as making a treatment plan early. This also brings up personalized medicine, which is medicine that is made for you based on your genetics. And the limitation that I chose to talk about was that there's little to no genetic diversity in GWAS. So a study was done in 2009 where they showed that 96% of the study groups were of European descent, which basically means that there is no representation for minorities in GWAS. And that's detrimental because the medicine isn't being made to cater for them. The fourth section of my paper was the future of GWAS, and I read a few articles on the future of GWAS. My first one that I read about was a breast cancer article, and it talked about how they could improve the tailored screening process for breast cancer to become more efficient and accurate. And to do that, they would use polygenic risk scores, which explain the relative risk of developing the disease and SNPs. And then the other article I read was about GWAS becoming available to the public. And a man named Valentine Heidert made an app called the Falcon and Shiny app, which is an app. It's an app available to the public where you can access genetic information. And this is very important because more people can become informed on genetics. So what's next for GWAS? The next step would be to become more involved in healthcare. Due to its many clinical applications, it's very helpful in disease prevention. This is talking about personalized medicine again because you can look for rare or difficult cases that don't have much prior knowledge on before. And if I were a researcher, I would want to expand genetic inclusivity and raise awareness on the situation with diversity. I would want to do minority-only based GWAS studies so that the medicine is going to become catered to them. And what's next for me? I'm referring to the Ohio Wesleyan University 2026 class. I will be majoring in biology with a minor in Spanish, and I will be going on the pre-med track so that I can go to medical school after undergrad. Thank you.
Hopefully you're still with us. We are halfway. Next up, hopefully something most of us have probably heard in the news. This was the 2020 Nobel Prize winner. But again, yes, next. Red piece. Red piece. Oh, that's the first one. All right.
Hello everyone. So for my senior year capstone project, I went ahead and did CRISPR-Cas9.
But first, a little bit about me. My name is Megan Rankus. I'm a level 2 biotechnology student at Woodridge High School in my home school, Theodore Roosevelt High School in Kent. Getting into CRISPR-Cas, so my original question that I set out to research was the future applications and current applications of the gene editing technology of CRISPR-Cas. Previous knowledge was in level 1, we learned about it just on a basic level, and then in level 2 we go a little bit more into depth and perform it in a lab setting. So my research methods, I had a mentor, Teddy Van Opsel, who's a PhD biological scientist. He focuses more on the clinical applications along with ethical challenges of biology and gene editing. So he helped me kind of figure out my sections, going more into germline and somatic editing and figuring out what it is for future generations. I used some books, biology books, which gave me definitions of gene editing, and then review articles. I used PubMed, which is a medical site that uses research articles that are peer-reviewed. So these are my sections that I used in my paper. I have my origin, which CRISPR comes from a defense mechanism in bacteria. How does it work? It cuts DNA and reconnects it by editing it through removing genes, adding genes, or just changing them. And then germline editing, which affects the future offspring and future generations. And then somatic, which is just affecting one single person. So my origin, where does CRISPR come from? CRISPR stands for clustered regularly interspaced short palindromic sequence. It originates in prokaryotes and bacteria. It originally is used as a defense mechanism in bacteria. And it's kind of similar to an immune response that humans have, where they detect virus bacteria or virus DNA, assimilate it, and then use it again as a future reference to destroy foreign DNA. And enzymes specify where it cuts. There's multiple different classifications of CRISPR. Who discovered it? So Jennifer Doudna and Emmanuel Charpentier. They're the 2020 Chemistry Nobel Prize winners. And they discovered CRISPR-Cas9 and developed it for humans in vitro in test tubes in the lab setting. How does it work? So CRISPR requires Cas9, a DNA repair system, guide RNA, and possible donor RNA. So Cas9 is an endonuclease, which is an enzyme that cuts polynucleotides. In this case, it's DNA, and that's why it's considered the molecular scissors. It requires guide RNA, which is a design component in a lab setting, and it directs Cas9 to a specific DNA site that a scientist wants to cut. There's two repair systems. There's homology-directed repair and non-homologous end joining. So HDR requires previous knowledge of the DNA that's being edited, just because it requires donor DNA, which is gRNA specific, so the guide RNA is within the donor DNA. And it has homology arms, so it's a more precise modification that matches the original DNA. And then there's NHEJ, which enzymes reconnect the cuts, which results in a little bit more unpredictable mutations. So this here is a diagram of DNA. So we have the genomic DNA going into the Cas9 enzyme. There's a PAN sequence, which stands for protospacer JSON motif, and it's just downstream of the guide RNA. You can see at the bottom there is the cut, and then there's a replacement sequence, so they decided to add something else in, and that's the repair there. So this is the repair systems. On the left, it shows NHEJ and the various mutations that come with it. On HDR, it's a little bit more of a simplified version. You can see the homology region right there, and it matches the donor template, so it's a little bit more of a seamless modification. So germline and somatic editing. Germline, the United States does not fund germline editing just because it can be manipulated and used for personal reasons. So the germline contains genetic material that gets passed down to offspring and future generations. It has effects, so these are the consequences that result from editing the genes. And then there's something called designer babies. So these are children that are theoretically edited in a lab setting, and you can put in or take out whatever genes you want, so they're customized to whatever you want as a parent. And obviously that's ethically immoral. So somatic editing is most likely to become FDA approved. It's any cell except for the reproductive cells, and it does not affect the offspring usually. And it's looked into for more for gene therapy. So sickle cell is a single gene inheritance. That is a limitation of CRISPR, is it only works on single gene inheritance, not multifactorial, just because it only edits out a certain gene.
And then it usually does not affect the offspring. In early studies, it did show that there might be a slight causation between gene editing and the offspring. There's no FDA-approved CRISPR-Cas mechanism for humans, so it's still being researched. What's next for CRISPR? So what I would do is I would do in-depth research into clinical trials, kind of go into more of a somatic gene editing. What else to learn? Laws regarding gene editing and ethical challenges. Since most legislatures don't allow for germline or embryonic editing, what would have to be in place in order for them to be able to do that? I would research human trials of gene therapy, as I said before, more somatic, and the off-target effects of germline and somatic editing is what I would study. Okay, what's next for me? I'm a biological science pre-med major at the University of Pittsburgh, and I wish to go into women's health. Thank you.
Okay, next up, we have Neva'eh Wilson. You gonna hold this? Wait, practice. Hold it.
Hello. So I research dissociative identity disorder. So a little about me first. My name is Neva'eh Wilson. I'm a level 2 student here at Woodridge High School in the biotechnology program. And I'm a senior at Quantum Falls High School, which is my home school. So an introduction to my prior knowledge and questions. I already knew a decent amount of the disorder and about the different personality aspects and the alters and everything. And my original questions were, is DID hereditary? Meaning, can it be passed down from parent to child? And are there any possible available treatments or cures in the works already available? So in my methods of research, I use a lot of research articles like Mayo Clinic, the Cleveland Clinic website, and PubMed that was mentioned earlier multiple times. And I watch a lot of documentaries and informational YouTube videos by Dissociative, and she talks about the struggles of DID and how the media and social perspectives can affect a person negatively with DID. And I use the DSM research book, which stands for the Diagnostic and Statistical Manual of Mental Health Disorders, which mainly has a bunch of mental health disorders and the medical information behind all of them. And lastly, my mentor, my beautiful mentor, Dr. Marissa Franco. She's a psychologist, author, and professor at the University of Maryland. And we talked earlier this year. She helped me out a lot. And she talked to me more about my future and everything, so she was really helpful as well. So my capstones, I'm going to go over all 4 topics later on, but I wrote this paper, and these are my main 4 sections that I'll be talking about a little bit. So what is dissociative identity disorder? I know a lot of you don't know what that is. Maybe you know it as multiple personality disorder. It's not called that anymore, it's called DID. And a lot of you might know it from the movie Split, which is not really a great perspective of how DID is in reality. So DID is basically a disorder characterized by the presence of 2 or more distinct personalities. And this is basically when a person's thoughts, memories, or feelings or sense of identity are disconnected from the host or themselves. So this impacts about 2% of the US population, and about 7% of people have DID-like symptoms but aren't diagnosed. This can either be because of lack of money to go to the hospital to get diagnosed, or they just don't go for multiple other reasons. So the causes of DID, so there are a lot of causes, but 90% of people with DID have developed it from experienced childhood or neglect. This can be anything from sexual, physical, or psychological abuse. And the other 10% of people have developed DID from trauma from natural disasters or some some medical illnesses or treatments that didn't go well, so they developed trauma from them. And DID mainly develops in early childhood and adolescence. This is not something that develops in your adulthood. So some signs and symptoms, these are the four most common. So significant memory loss from switching out from the host to the alter. So throughout the day when you're switching or dissociating between alters, you kind of lose track of your memory a little bit. Depression and anxiety, like I said earlier, this can come from a lot of media perspectives of DID and the way people perceive you when you have DID. Out-of-body experiences, so this would be like watching your own life play in front of you like a movie. Like you're at the movie theater and you're just watching your whole life play out in front of you and you can't really control anything. That is a big symptom and it affects a lot of people. And lack of self-identity, so you're not sure which alter or person you are because of the dissociative aspect. And current treatment options, there aren't very many, but DID is managed through various therapies like talk therapies and psychotherapies, which are the most effective. And managing other symptoms, like the symptoms I just talked about before, those ones are most managed because there's no cure for DID. And my discussion topics, there's still a lot to learn involving DID, like looking more at the brain scans of people with DID versus healthy patients, looking at the brain scans to see if there's any differences between them, and then the genetic variation between alters in the host, which I find most interesting and probably would research more into along with the subgroups of DID. But mainly genetic variations because other alters can have different allergies or different eyesight or vision and different handwriting or left or right-handedness. So I would probably research more into that because I find that personally very interesting. And what's next for me, I'm a child psychology major at the University of The Ohio State University at Mansfield, and I'm hoping to become a family or child psychologist.
Last but not least, we have some Woodridge representation. Is that just the screen here? You can't see that? Bear with me. There we go.
Hello everyone, nice to meet you. My name is Fatima and I'm doing a research project on Alzheimer's disease. So a little about me. I'm a student at Woodridge High School and I'm part of the biotechnology program. I'm also part of the National Honor Society here. So the reason I'm studying Alzheimer's is because I'm largely fascinated by neuroscience and how the brain works. And with Alzheimer's, I wanted to learn more about disease that's affecting many people around the world. And so I thought it would be a good way to delve into its consequences as well as its genetic components to learn more about why it does the things it does. So my mentor for my project was Dr. Mabel Terminal. She's an AAES Science and Technology Policy Fellow for the National Institute of Health, and she studied at Texas A&M. She currently studies neuroscience and is researching Parkinson's disease. So my research methods for the project were my mentor. She mainly helped me understand the neurological aspects of the disease as well as the genetic components. And I also got articles from her and articles from the research database PubMed. So what is Alzheimer's? Alzheimer's is a disease that affects basically your cognitive abilities, such as memory, speech, communication. And it's basically caused by the buildup of beta amyloid proteins. And that really causes plaques to build in the brain. And when that happens, it basically prevents the neurons from communicating with each other, which later pushes on towards the symptoms, which includes memory loss and communication issues. So Alzheimer's has different stages. The first stage of Alzheimer's is the preclinical stage. During the preclinical stage, patients don't usually have much common symptoms. That's why a lot of people don't even know they have Alzheimer's during the stage. The next stage is the middle stage. Now during this stage, a lot of the symptoms become more prevalent. People experience memory loss or communication issues. And it isn't until the late stage where the symptoms become more and more prevalent, where people have difficulty socializing and even just interacting with others. So the origins of Alzheimer's. Alzheimer's was originally discovered in August Dieter, and while she was the first patient to actually have Alzheimer's discovered in her, I wouldn't say she was the first patient to ever have the disease. It was just during that time, which was the early 1900s, specifically 1906 in Germany, where people finally learned what the disease was. Now, she was originally a housewife, and she had difficulty with her memory at first, and she had difficulty doing her basic chores. And so her husband took her to the hospital later on as her symptoms progressed and got worse. And she was put under the care of Alois Alzheimer, her doctor. Now, Alois Alzheimer originally diagnosed her with regular dementia, but it wasn't until later on where after she died and he took her brain, he got to test it more and do more scans where he diagnosed her with Alzheimer's. And what's interesting about this case though is because a bunch of other scientists at that time also discovered the disease. But the professor under Alzheimer's basically named him the head of the whole disease. And that caused like outrage towards other scientists obviously because it wasn't really fair that he got the credit for discovering something that they all did. So the genetic components. So there are many genes involved in the disease, but the most prevalent gene is the APOE4, which stands for apolipoprotein E in allele 4. Now this gene is a risk factor gene and basically enhances the possibilities of you getting the disease. Now many people have the gene, so it doesn't necessarily mean you will get the disease. But what's complicated about this is because there's very limited information, so not many people know why this happens. And so there's not much information to see what causes APOE4 to give people Alzheimer's. And another thing that may cause the disease is the tau protein. The tau protein is a microtubule-associated protein. And microtubules are basically proteins that transport nutrients in the brain. And they help the brain towards cell signaling and communication. And so when abnormal tau proteins occur, it causes them to clump together like cancer cell tumors. And when that happens, it disrupts communication, which pushes towards Alzheimer's disease. So the current main treatment for Alzheimer's is aducanumab. Sorry, I can barely pronounce this word. And it basically is the only disease-modifying drug approved by the FDA. Now, with this drug, it basically attacks the beta amyloid plaques. But what's unfortunate is that it only works during the preclinical stage. And like I said before, many people don't really show heavy symptoms during that stage. And so a lot of people don't even know they have Alzheimer's. And so the drug is in fact effectively useless. Now these are the other drugs, which is memantine and donepezil. Memantine modulates glutamate, which is a neurotransmitter, and donepezil, it prevents the breakdown of acetylcholine. Now both these neurotransmitters push towards cell signaling and communication. And if they're both disrupted in the brain, it can cause brain death. And it can cause just the brain to stop, like the neurons to stop communicating with each other. So what would I do next? Well, I'd like to research more on the other types of genes associated with Alzheimer's, as well as how they might affect different patients. And what else is there to learn? Maybe like with the drugs, I'd like to also see how they might affect Alzheimer's in its different stages, because currently the drugs only work during the preclinical stage. But it would be interesting to see what damage or what breakthroughs they can bring towards other stages of Alzheimer's. And what study would you set up next? Well, I'd like to study like other diseases that deal with like brain issues or brain diseases similar to Alzheimer's, like Parkinson's. And what's next? So after high school, I'll be attending Case Western Reserve University, and I'll be double majoring in biochemistry and psychology. I'll also be on the pre-med track and hope to become a doctor in the future. Thank you for listening.
All right, well, we made it. Thank you all for coming and listening. I think we did a really good job. Especially, I don't know how long it's been since some of you saw like a 15-minute science lecture. But, you know, I tried to keep it short. I tried to keep it under an hour. I'll have 12 in a couple of years, and that will be interesting as well. But regardless, I really hope you guys understand why I'm so proud of these guys. And obviously, hopefully you guys learned something. Perfect timing, I guess. Anyway, but yes, so I'm excited to see where these guys go and hear from them the next couple of years, the next 5-6 years from now, and especially see why they're have really good reason to be proud of themselves and you all to be proud of them. So this night was for them. I don't want you guys to rush off.